Hi, my name is Ken Lassenson. I wrote the Microbiome Prescription website, and today I'm doing a walkthrough for new users of Biomesight UK Microbiome Testing 1, showing how to transfer the data over to my site, do analysis of it, etc. So this is a simple, straightforward introduction intended for new users of Biomesight, to make your life a little bit less stressful. Okay, you logged on to their site and you get a whole bunch of choices here. And we have external apps here, which is what you want to look for. Click Microbiome Prescription. And you go down and you select a sample. You notice that we have some labs which are not Biomesight listed there, and that's because Biomesight, for free of charge, provides translation software for other sites' raw data files, which are called FASTQ files. So they will shove through their data and convert it to the same standards as they're using on their site using the raw RNA data, which is very important if you're doing comparison from one sample to the next. You want them to be processed through the same software. If you use different software, it becomes a apple to watermelon problem. So they provide the facilities to make your life easier and to allow direct apple-to-apple comparison. So you select the site you want to send over. I'll go and do one from August. Click here and the transfer will start, and you will get a notification in a few seconds up on screen that it is completed. Then it will automatically send an email to you of how to log in. And let's take a look at one of the emails. Here's what the email said. It has a link here. This link will automatically log you in to the website. Nice and simple. And so all you need to do is just click it. Directs you to the website. Let's finish bouncing around. You know, once you get there, you have a choice of automatically going to the sample page or going to this page or not. Some people would like to save this page up here as a favorite so that you can do it. You can reset the login whenever you want to for your own security. So you could if you wish, share it to somebody else and then terminate the sharing by simply resetting the password. So let's just go into looking at the samples. We have one sample transferred across, and okay, that's I'll need to come back to. So changing your microbiome site, and we go down and we have a bunch of choices for changing a site here. You could, for example, want to do a quick health analysis, and we see how you are sitting there, percentile ranking for things, and basically you have no problems there. You have Jason's Horlock's suggestions and which are ideal, which are not ideal. You have a listing of bacteria which are healthy or not unhealthy, and as to why everything is provided by citation as to where the information is coming from. In this case, there is nothing which is particularly bad except for one thing, which is a bit of concern. It's connected with fat percentage and blood pressure, and if you want to know how we're getting this data, we get it from the study, and you can actually go over and read the studies and see how things are connected to it, and you can see the name of what it is. So we have provide the information with information sources. So you do not trust me. I don't want people to trust me. I want people to say, look, here's the source. You can agree or disagree as you wish. So that's basic thing. Go back to 16 samples. Usually the first thing that people want to do is improve their microbiome. And that's all we look at this time. Now I have here nice simple choices. We have a small number of choices here. Up here we have display level, if I click it over to advanced and we go back and look at the choices, we see a whole lot more things. And the reason we have layers of options is because, to be frank, there's too much information available for a first-time user. So we'll go back and we'll just mark it as a beginner. And now you go in here, and we will usually be using suggestions using expert criteria. And there we have, well, people have different beliefs as to what a healthy microbiome, or has different mechanism to calculate what is abnormal or not abnormal. So we have here, we have ranges from a variety of labs or sites, which I have obtained from their reports. We also have ranges calculated using the standard methodology that labs use, which is what some of the above labs use and some of them don't. Something called box whisper, which is a method which basically calculates the ranges, and we detect anything which are beyond the riskers, which means they are abnormal or deemed from data science to be abnormal value. Cutoff Motrop is a proprietary algorithm which I have permission to use, and which does some curve filtering, and that goes off and explain. Or you can say, okay, show me based on everything where I have at the top or the bottom 10%, where I have more extreme values, and you can adjust that to whatever percentage you want. So let's walk through them quickly. I'm going to click on JSON, and the thing you will notice is open up a new window. So you can do side comparison, and we see there, and we see we have 8 bacteria selected, and the ones are here, and these are the ones which are either too high or too low. And in some cases it will give information whether or not a pathogen, and again links to the medical literature for it. So that's the first one. I'm going to go back and say, okay, let's use this one. Here is a lab with 54 criteria. How well do we do there? And we get 8 again, and guess what? They look very similar to the first 8. Sometimes there may be a change of 1 or 2. Most of the labs have come to sort of a bit of a agreement as to what is or isn't. Here again we have 8, and we have similar there. Although we have 59 criteria, you only are outside that range, the other range, for 8 of the 59. Number 1, Nirvana. Again we have magical 8, same as before. And Xenogen, the last time, I expect 8 for the same reason. We have 8. And so these are the major culprits that these labs find in common for most people there. However, that doesn't necessarily mean that's a cause. These are simply what everybody agrees are major indicators of issues. Not minor, but major. Now we can go over down here to a standard lab report. And here it explains the methodology of it. And I'll click here instead of clicking the informational link. And we have only 4 which are outside the range, and they are totally different. And we have suggestions down below. Interesting. Now we go to Box Whisper. And again, I click the information page rather than the box. Let's see what comes up there. We end up with 34, a lot more. So we see it's clear there are disagreements between methodologies, which is not unexpected in science because we're dealing with scientific theory, not demonstrated fact. Last time, let's do Cutoff Motrop. And we have 69. So at this point of time, let's do last one, and let's do top 10%. Let's see what happens there. 77. Oh boy, we have a lot of bacteria selected, and you can see all the bacteria here and which ones you are in the top or bottom 10%. We could drop it down to saying, okay, let's just do 1%. and we have exactly one bacteria where you are at the extreme. Now for each and any one of these bacteria, you do have the ability to just click here and it will take you over to information page, and information page shows what is associated with it, whether or not it's associated with, for example, high colorectal cancer, or if you have low with hay fever. So again, everything is connected to citations. But let's go back to keep this simple, and we have a bunch of suggestions. So the first question is, okay, you're tossing at me 10 different sets of suggestions. How am I supposed to figure that out? Well, I listened to you, and if you go back to the 16 sample page, you will see there's something called consensus suggestions. Every single suggestion you run gets added into a rolling collection of suggestions for the last 24 hours. You can go and you can get rid of all of them if you want, or you can go and view the consensus, or you can do list consensus suggestions, and it says how many suggestions, how many items came from the suggestions. Remember the first ones where we had almost all the same bacteria? Guess what? We had the same suggestions, and actually we only showed about 20 ones on screen. There's 375 suggestions underneath the cover there, and the last one we had just 20 suggestions total. So we can see what we've done and how dense variety of things are. But in general, what you want to do is go down and view the consensus. And the consensus will go through and list out things there, and it does it in layers. Things which are the absolute safest, things which all the ones actually agree about or has no comments about. In other words, these are the safest things to do, which has a probability of improving your gut bacteria. So you go down here and you can pre-filter them. For example, if you wanted to look at vitamins only, or here's a combination of those. One item you find selenium as a supplement. You find that it is pretty much consistently suggested by 8 different of those 10 suggestions says selenium is help. Go down to the next one, and safer takes, which at least one of the 10 things we ran says, not so sure about this. Slight degree of hesitancy, but you can go down and take a look at what they are. Aniseeds, almond skins, a particular probiotic. And now you hit the likely safe, which are things which are, you have the pros and cons. 1 says yes, 5 says no, but the overall net value seems to be positive. And then you get your highest risk. These are things which you should try to eliminate from your diet totally if you can. And we have it down there, 75 items there. Ones which are not as extreme, but you may want to consider trying to reduce them. For example, a low-fat diet, and one who has some adverse one. For example, some probiotics products may not necessarily be best. So this gives you a consensus. My general suggestion, first time through, look at your safest takes. Go through and take a look at what they are. Use the buttons up here to quickly sort out. Things are there. Notice that the columns all sort there. For example, chamomile or chamomile tea is a take on 9 out of 10. In other words, everybody says take chamomile tea. May not have a great effect, but will have a positive effect for each one of those 10 very different selections. It's something which is being held in common by all of them. So it is a good thing to take. Ginger, peppermint tea, etc. So filter as you want. So that gives you how you deal with information overload from the suggestions. You go run them, check the consensus, and work from there, so you have the best of a difference of opinion. Now, if you go back here, let's go back here, and we have the the health analysis, which is a quick look. Biomesight does a good job of it. This is simply a alternative. And it basically says you have three bacteria associated with a healthy individual, and you have 10 which are unhealthy. And it's right here. You find the study from which the information is obtained from. We are constantly adding studies every week, literally every week. I review the latest studies published in the last week. So there are conditions there. You have no apparent, or actually it looks more like I have broken something, which I will fix. Probable medical conditions. These are items which you have probable. Generally, things like Parkinson's disease, etc., are rare things and no particular issues. I do need to update that chart. I have added more information. One of my joys is playing catch up. If you go and click here, it will take a chart and it will show you which bacteria are involved, whether or not there's agreement or disagreement, your value in the percentile. So this gives you a detailed breakdown, and as always, link to the studies where I get the information from. I present every study, put in there, even if I disagree with the result, or if 8 says one thing and 1 says the other, I put all 9 of them in there, because I want data to be totally open data source. No black box, no trust me, no I'm an expert. I'll give you the research and you can make your own decision whether or not my conclusions, or the should say not my conclusions, the AI conclusions are reasonable. Then we had Jason, who I have attended lectures with. I talked with him and gives consensus, ideal and not ideal. You remember Jason H was one of the choices which we did. And then we have the unhealthy bacteria and which ones are unhealthy, the possible conditions associated with it, and you can click citations to find out the study where the information comes from. So again, everything is open. Okay, now I'm trying to keep this short, and let's go back, and we go back here. I'm going to do one more thing, and I want to increase this one a little bit. I want to take you up to intermediate, because fingers crossed, yes, everything which we have dealt with there comes directly from a PubMed study showing just substance modified just bacteria, and I've put together all this data that I could find, and that's a large number of items, literally several million. But there's a second way of doing it, because that was dependent on clinical studies being done. Fortunately, there's a site called KEGG, which is the Kyoto Encyclopedia of Gene Genomics, KGG, and it has done DNA level analysis of different bacteria and which enzymes to produce are in each bacteria and what products they produce or consume. So from that information, I have built a little AI engine which goes and calculates how much you're producing for everything, what you're short of, and is there a way of supplementing what your microbiome is not producing enough of, as a way of help normalizing. If your body isn't producing enough of a particular enzyme and you take a probiotic producing that enzyme, conceptually that will help eventually balance your body, because it will make the environment more normal, more healthy, and then eventually things settle in. So we have two things. First of all, we have AI suggested probiotics, which comes here, and it's ranked order as the one which has the greatest amount of impact. And go down and take it. For example, this one, Miracin, happens to be one which I personally take a fair amount of. Alpantarian, I happen to be doing right now. The pendulum, I take part of it. I take the Acromantia, and it's Symbiphore 1. Yes, I take that one. So it gives you a list of the probiotics there, and there it limits you to the top 60. You'll notice some says ES or DE indicators this is the country where it usually is obtained from easily. Others are a bit of a challenge. Here's one which is a German one. You may not be available outside of Germany. Mary region Japan is available in most countries, and Amazon is a good spot for it. So that gives you a list of probiotics which are determined strictly by the computed amount of enzymes, several thousand enzymes, the different that your body bacteria produces, and looking at what the probiotic bacteria produces and complements. And this one is one which I must admit I'm most excited from. It's a nightmare to get the calculations done, as in literally hammering a PC at 100% CPU for literally a couple of days. But it's done, and it gets updated periodically about once every 3 months, just in case something has changed. Let's go back and look at the other one. The other one is a little bit more restrictive because not everything is available as a supplement. So it states two things: beta-alanine, which is available as a supplement, Microbiome, which is also available, and according to our computations, you are at the bottom 5.7 percentile of items. Here we are, because of variety, some people at 10% will get a list of 14 items. Other people at 25% will get almost nothing. Depends on how well balanced your bacteria is. So let's go up to 15%. And we'll get some more things added like magnesium, glycerin, beta-alanine, NADH, all of there. That's percentile. So you can use that for ranking. The ones which are the lowest values are likely the ones you should supplement first, and go from there. So that's basically it. Let's go back, and there are a bunch of other things available. There's symptoms, which is I've done separate videos on it. Let's not go there. We have component analysis. Component analysis gives you the details. Again, it is, let's flip back to beginner's level. So we have less. It gives you a variety of what bacteria you have, what end products are being produced, what for example, you click here, let's just go there, and I may, key thing, I have nothing there, and I given just two choices, nothing there. But if I go up to intermediate or advanced, I can go back to end products. I have the choice of seeing all items. Oh, dry. Okay. Custom percentile. Oh. Let's go back. I think I know what may have happened. Okay, and okay, you can look at the end product, and you need to upgrade it to probably intermediate. Sorry for the bouncing back and around. That's a percentile. And there, if I jack it up to 10 percentile, you see what you are at the bottom or the top. As in, for example, testosterone, your production level seems to be higher than normal. Well, hopefully you're a male, but if not, well, no comment. Niacin, you have high production of it, which implies you probably do not need niacin as a supplement, etc. Here is a supplement which you may be able to find as in one form or another as a supplement. And up at the top it says that one of the questions is this really statistically significant or not? We would expect 33 items to somewhere between 0 and 33 items if you are really out of whack. We don't have that number. Generally, it's probably not worthwhile doing things too much. However, that's a personal choice or choice you make with your MD. So that's their component analysis. I like to generally show people, sample visualization, my biome view, because what it does is it shows the hierarchy of bacteria. You're talking about species or parts of genus, parts of a class, parts of phylum, and it shows the structure here, and then shows you where your numbers are out, and it highlights the ones which are of concern there by color, high or low, and it shows you all your bacteria in the structure, which is helpful. If you become an advanced user, you can go and actually start hand picking the bacteria you want to use for your suggestions rather than a canned suggestions. Okay, at this point in time, I'm feeling I'm probably saturating people too far, so I'm going to basically end here. I have lots of videos taking a few different parts of the site, and also see a couple of bugs which hopefully will be fixed shortly, and that's it.